Good morning, everybody. Good morning to our friends from France, and good afternoon to our friends from India. Welcome to the second day of the workshop on AI for healthcare. Uh, I just want to, before I talk about the program, I would like to request my co-host, Professor Jan from France, and request him to introduce himself briefly. Jan, would you please introduce yourself?
Sure, thank you. Thank you, Nikhil. I'm Jan Bunel. I'm a professor at IMT Atlantic, which is a technical university in the west part of France, spread on 3 campuses in Brest, Nantes, and Rennes. And I'm the head of the department that represents all the activities of the Rennes campus about system network, cybersecurity, and digital law, and how we use the artificial intelligence in this kind of solution. I'm very glad to be here and to co-animate this workshop with Nikhil Pal.
Thank you, Ian. Actually, you did a very good job, but I did a bad job. I didn't say who am I. So you have seen, my name is Nikhil Pal. I'm from Indian Statistical Institute. We have a center for AI and machine learning, and currently I'm heading that center. So that's a very brief introduction about me. Now, before we go to today's session, let me quickly say what happened yesterday. Yesterday, we had very interesting 8 presentations, and these presentations were from different topics, 4 from France and 4 from India. Like the topics covered were prediction of drug prognosis with multimodal data, binding site prediction with AI, better representation of data for better AI applications, few-shot learning for neuroimaging. That is also related to data again, because you don't have too much of data there. Applications of biomedical image analysis, including non-invasive monitoring of cancer biology. There were discussion on several AI-based real-life applications to healthcare, including prediction of hemodynamic shocks, issues with real-life medical data, because success of any healthcare AI application depends on data and good data, trustworthy data. So we had very good discussion on issues with real-life medical data, how to deal with that, representation learning for affordable healthcare, and ML for better and low-cost COVID-19 diagnosis, and such tools probably can be used for other emergencies also. AI for pharmacovigilance, and many more. So it was fantastic sessions. And we have today also several very interesting talks. In fact, we have 6 talks, and they are grouped into 3 segments. The first segment has 3 talks, then we have 2 talks in the next segment, and the last segment has only 1 talk. Now, each talk is of duration 15 minutes, with the understanding that you will finish your presentation possibly within 13 minutes. Doesn't matter if it go to 14 or so. We kept some time to switch over to the next speaker. And those who were present yesterday, they know that we don't have Q&A session after every talk. Rather, after every segment of talk, we have Q&A session. For example, after the 3 talks, we have 20 minutes slot for question and answer. So I would request every speaker of the segments to remain present till the Q&A sessions are over. In fact, I would request them to present till the end of the session. So we shall follow the same protocol today. As we may take few pictures of the participants in between. And finally, and it's very important, the day ends with a general discussion to arrive at some conclusion or suggestions, maybe actionable items that can benefit both countries. So I request, please be present if you can till the end of the session. So with this, I will finish my introductory remarks, and then I will I'll move to the first talk of the day. And the first talk is by Professor Mithali Mukherjee from Indian Institute of Technology, Jodhpur. She is currently the head of the Department of Bioscience and Bioengineering. Professor Mukherjee, if you are there, please share your screen and you may start presentation. I don't see Professor Mukherjee. I don't see Professor Mukherjee. Then, Jan, should we switch the talk? What is the time now?
Yeah.
We have still 3 minutes to start, so we can wait for 3 minutes.
Yeah, maybe the speaker will just appear. We hope so.
Yeah, because it's online, so 3 minutes good enough time to join.
Hello, this
Yeah.
is Debarco from Delhi, and Professor Pal. I just had a word with Professor Mitali,
Mhm.
and a few minutes back. So she is supposed to join, I guess. So let's wait for her.
Thank you. Thank you, Debarka, for this update. It gives a great relief.
Thank you. So let's wait for a couple of minutes before her arrival. Thank you very much, Demaka.
So, Jan, how is the temperature like in your place?
It's quite cold. This morning it's 2 degrees when I woke up. 2 degrees.
Oh, my goodness. Calcutta is very comfortable.
Yeah. I have a good heater at home.
Okay.
We are used to temperature in the winter in France,
But it doesn't go to negative, does it go?
Yeah. A couple of times during the winter we go to -2, -5, especially in the night.
Oh. So the best winter time is yet to come for you.
Exactly.
And Debarka, how about the temperature in Delhi now?
Sorry, am I audible, Professor Pal?
Yes, you are audible.
Yeah. So Delhi is also pleasant. It's just that Delhi has a very bad pollution these days. And Delhi temperature oscillates between, I guess, 13, 14 degree or 10 degree at midnight to 22 to 25 degrees in the morning, in the daytime. And by the way, I was in Nantes just before the COVID came, right? Nantes, I think Ian is from Nantes, is it?
Yeah, I'm from Rennes. There is a mistake, but yeah, it's one.
Okay.
So, but we have a campus in Nantes. That's true.
Yeah, Nantes, I was in this hospital, and I have a collaborator there working on infectious disease, and I sort of collaborate with them on AI support, and it's a very nice place.
I'm agree. The west part of France is a very good place for living. And Ian, who will be the next speaker just after for the second talk, cannot say something different, I think, because he is also from Rennes.
So, for the interest of time, and if I'm seeking your suggestion. For the interest of time, should we switch with Professor Lecomte? If Professor Lecomte agrees.
Or we can as well switch with the third talk so that we are still one French, one from India. It's up to you.
Okay.
That could be interesting. Yeah. Thanks for the proposal, Ian.
Yeah, that's okay. So with Debarko, Professor Debarko Shengupta. Debarko, is it okay with you?
It is almost okay. I just need about 2-3 minutes, if that is permissible.
Well, we have to because it comes suddenly. So by these 2-3 minutes, if Professor Mukherjee joins, then of course we are done.
Exactly.
Or I can start if you prefer. I mean, it's real to you. I was just suggesting for the sake of having one-on-one, but you let me know.
Yeah, I think Professor Lecomte, if you are okay with this, please start. Are you okay? Okay.
Yeah, sure.
So, friends, as you have seen that the first speaker has not yet joined, so we are making little change here. So our second speaker, Professor Jan LeCun, who is from University Rennes Irisa. So he will be giving the talk first. The title of his talk is Harnessing Prior Knowledge to Improve AI Algorithms for Patients Representation and Classification.
Can you see my screen
So,
properly?
yes, but maybe you have to go to full screen because
Oh, okay.
it's,
So I am in full screen, but it's not showing the right screen, I think.
I think now it is okay.
But now you see just the PowerPoint, right? Not the full presentation, though.
Full presentation shows up on the left side, and
Yeah, I'll try to switch once again in screen mode. But do you see the speaker mode or the full screen?
no, we don't see full screen. We see the whole. We see the full screen really.
Okay, and you see the screen. This is changing right now.
It's changing.
Okay, perfect. Okay, so then let me start. All right. So thank you for the invitation, and I'm happy to fill in the spot for the last minute in the morning. No problem with that. So just first, I'll start with a word about what we do in the team, which is called Delis, and basically, just very briefly. Historically, the team is about more bioinformatics and retrieving large amount of data from the online databases, which could be semantic web, ontologies, annotations, or even gene expression, then structuring this knowledge and extracting models to describe a biological system, and then providing tools for experts to navigate into all these models. And for the past couple of years, since I joined the team, we have been interested in how we can get this structural knowledge and plug this knowledge into AI algorithms to make them better, having better performance, better predictions, and more interpretability. And this is on this last part that we're going to focus on today very briefly. And one thing that could be interesting for us in AI in healthcare is all about representation learning. And just an illustration briefly that you could have from multimedia data set. If you have a document that is described by an image and a text, like in orange, for instance, here, I don't know if you see the pointer. What you want to have is to project this multimodal document into a single subspace and do this for all the documents. And in this subspace, what you expect is that similar documents will be clustering together. And in terms of application to AI, then you expect that you have a subspace of patients that have the same disease, as we saw yesterday for a couple of examples, for instance. And the context for this representation learning, when it becomes interesting, is when you have multimodal data. Each document or each patient is described by different type of data. Sometimes could be multi-view, like you could have multiple images for given patients. And it works very well when you have very large data sets to train the models. And of course, we want to know whether this could be applied for healthcare, because patients are very often described by this kind of data. But basically, there are a couple of challenges here. And as Professor Pratosh mentioned yesterday, very accurately, one of the big caveats is the small data sets. And for instance, in Rennes, we have a cohort of Crohn's disease patients, and we are measuring the microbiota of these patients to try to predict the evolution of the disease. And we have about 300 patients, which is already a huge cohort for this specific disease, but we are nowhere near the millions that you have in multimedia data sets. And of course, there is the need for interpretability for the patients to understand the diagnosis, or for the doctors to react accordingly. And of course, there is a last problem, or let's say challenge, that is about variability between patients and in time, even for the same patients. So what can we do with small data sets, basically? What we try to do, and I'm going to give example later on, is to harness what transfer learning can do, that is retrieve a model trained on similar problems and then just fine tune it on your small data set, and it's worked quite well. We relied also on completely unsupervised method, that is we don't need any labels. And finally, this is the main point of today, we try to retrieve other knowledge to enrich the data sets to better describe the patients and see if we can cluster them even better than before. So basically, can we get what bioinformatics knows how to do, that is retrieve databases, structure knowledge, and put this into AI, basically. An example from a paper that was recently published on Microbiota as well, Microbiota data. What they did is that they took, so I don't know if everyone knows what microbiota is. It's basically all the species that are in the gut of the patients, basically, and you count how many how many individuals, how many bacteria you have of each type, basically. And from this, you can project the data. So in the paper, they use an autoencoder, which is a classic neural network architecture. But what you get is a compressed representation of each patient. And from these compressed representations, they plugged in classifier, usual classifier for machine learning. And what they showed very briefly is the result in red is that their approach was outperforming a different classical machine learning approach, that is projecting data and then classifying seems to improve prognosis for patients. And we use the same kind of approach on our cohort for Crohn's disease patients. We went to normalize the microbiota data, went for unsupervised clustering, went for different kind of projections. And very briefly, what we showed is that each dot is a patient. Each color is a group subtypes of microbiota from the unsupervised clustering. And we linked this to the medical data. And we showed actually that from left to right, you go from mild symptoms to very heavy symptoms. That is, the projection, the representation learning seems to have already quite an interesting properties in terms of predicting the heaviness of the symptoms, of the gravity of the symptoms, if you want. And from there, we want to know whether if we add more knowledge to the process, can we improve even more this representation of the patients and predict even better what's going to happen for them. And to give you an idea of what could be done, and microbiota is very well suited example for that. So this is a paper, and I'm going to drive you through this figure, which is a bit big, which has been published from the team last year. And basically, you start from a list of microorganisms from an ecosystem, and could be exactly a microbiota, for instance. And from this list, we developed a pipeline that is going to query databases to retrieve all the genomes for each of the different organisms that are in the mix. From these genomes, we retrieve all from these sequences, right, we retrieve all the annotations, the functional annotations of these sequences, what are the possible metabolites in the system. Then we automatically rebuild metabolic network, how these metabolites interact with each other to provide higher level functions. And then finally, because all these species are in the same ecosystem, and maybe you could see on the bottom part of the screen here, you see that if a network is producing a metabolite yellow here, this can be used as an input for another network. So basically, you build kind of a meta network, and it tells you what the community as a whole is capable of doing in terms of metabolic processes. Okay, and this information is very interesting because we can plug it into our AI system. And let me give you an example. So you start from this very simple table, which is basically patients in rows, and what you see S1, SP are the different species that are present in the different patients' guts. And the number here are the count of each of the species, okay? And with a tweaked pipeline, but very close to the one I just described you, what we're getting is that we're getting the proteomes for each of the species, for S1, for S2, and so on. We cluster the sequences and we get annotations. That is, we get the main annotations for each of the species. And then the next part is that we combine this count of patients on each species and the information we get on each species to change the description of the patients. And we don't have just a count of patients per species, but we have a count of functions per patient. So we change the description of the way we consider the patients by basically reaching out to prior knowledge about the species and about their sequences. And so far, what's interesting is that if you change the representation of the patients and you plug traditional machine learning classifier, exactly like in the paper I showed you earlier, we're reaching the same level of performance, but what we gain is much better interpretability. That is, we can predict that each patient has a given disease depending on the example, but not only because he has species 1, 10, and 20, which can be sometimes a bit cryptic to understand, but by saying because there are these functions and these functions that are overrepresented or underrepresented. So here, performance are similar, but we gain a lot of interpretability by having this extra knowledge. And this is just like actually a project that we started a couple of months ago, actually 2 months ago. So it's already very promising. And we're still working on this right part of the screen, how you go from basically, and this is what I wanted to show on the right, the biggest problem is when you retrieve knowledge in terms of healthcare, usually the knowledge is on the descriptors, like here on the species. And you have to go from item-based knowledge to patient-based knowledge. How you go, from the description of the columns to the description of the patients. And this, we're trying different methods to do it. And each try we do are improving actually quite dramatically the prediction for the machine learning thing. So there are plenty of things to do here. And then on the long term, we want to see if we can have a smoother way to integrate the information, that is having different kind of information on the sides and talk to each other, let's say. That is the very classical multimodal data integration problem. And we can add other types of knowledge by interrogating databases, for instance, about the different species and the disease they are involved in, or the papers about this disease, this species, sorry. And this basically raises the question as well of how much information should we put, how much external information should we put, and will we be diluting the initial information? So it's all open questions, like theoretical questions, but are of interest for us. In 2 minutes, just a second example, because I think it was also of interest for people from yesterday. We started another project a year ago, which is about, and you start from the bottom here. You take a sequence of amino acids from a given proteins that you see here, and you use neural networks that are typically developed for natural language processing that are called transformers. And this network will provide you with an embedding of each of the token, the projection, if you want, of each of the token. And from these projections, then what we do is we derive a prediction for the protein function or the domain function. And when we compared the model we developed with other state-of-the-art model on 2 data sets, we're quite above what was previously done. And the model is called my model because the students did not come with a fancy name so far with what he developed. But on top of this quality of predictions, what we're really interested in, is the interpretability. And what we could get from our architecture is a highlight of the different spots in the sequence that are very important for the prediction of the function of the protein. And here's just an illustration, but just to give you an idea. On the left, you could have the color that is linked to the importance of each of the position in the proteins for the prediction. And on the right side, you could have a highlight of known biological properties that we know from other databases, that this part is important, this part is important. And then we want to see how much overlap we have between what we think is important for predictions of the function and what is important for other biological function. And what we did actually, and this is not a full picture, but it's just to give you an idea, but what the student did is to benchmark a lot of interpretability methods and measure every time whether what the method thinks is important for prediction, does it fit with known biological properties? So basically, it's kind of an interpretability of interpretability methods. Basically, the question is, which of these methods seems to be more suited for studying sequences of proteins in terms of understanding or finding new biological properties? And actually, what's interesting, and I'm not going to dive into too much details, but actually the attention-based methods seems to be performing super well and are really linked to the architecture we have. So I think it's very promising. Just 2 words to be in the 14 minutes range in terms of prospects and potential collaborations. And this is not exhaustive, so feel free to discuss this with us, of course. Basically, if you have a machine learning AI problems and you know there are external sources of data, of knowledge on your data set that could be used, then we have all the bioinformatics part of the team that is interested in all the challenges of retrieving data, structuring data, and so on. And especially when it comes to chain queries and semantic web, we have also a lot of things on these aspects. And finally, there is this very AI-specific question that is, if you retrieve knowledge, if it's a graph, if it's an image, if it's depending on the structure, how do you include this into classical machine learning algorithm, which is a very theoretical question, but very interesting for us. And last but not least, and it was a bit mentioned yesterday as well, we also developed in Rennes over the past 8 years a specific curriculum designed to teach AI and applied AIs to non-specialists, and especially for biologists and doctors. And we'd be very happy to share as well what we tried to put in motion in Rennes, if you're interested in these aspects as well, because I think if we want AI to get more traction, at least in hospitals or in very, where it could be very used, I think we need also to think about the training programs that goes with it. And that will conclude the presentation for today. And thank you for your attention.
Thank you, Professor Lecunf. You have done a fantastic presentation and right on time. In fact, we have enough time to switch over. Thank you very much. We will not go for Q&A now, as you know, so please bear with us.
Thank you very much.
Professor Mitali Mukherjee, are you there?
Yeah, I am extremely sorry. I missed the time for 2:30 and I logged in thinking I've logged in.
Oh, I got it. Yesterday it was 2:30. I see. So no problem. So
Maybe
let me.
Barka can present before and then I'll go in. I think that would
Yeah,
be
that's fine,
good.
Professor Mukherjee. I can present if you wish.
Yes.
Okay. Friends, there is again little more change. So, Professor Debarka Sengupta is going to make the next presentation. He is from the Indraprasth Institute of Information Technology, Delhi, and he is going to talk about gene expression based inference of cancer drug resistance. Very
Yes.
interesting topic.
Am I
Professor
audible to
Debarka,
everybody?
yes, you are audible. You are
And
audible.
your
Maybe
screen
you
is,
can make it full screen.
I have made it full screen. Is that visible?
It's visible, but maybe I should try to. Maybe it's visible to me, but it appears small. So that may be a problem with my system. Maybe you
No,
can
no,
go
no.
ahead.
We also see it as a it's not in the presentation mode, Vivarka. So
Yes, see, okay. So now also you can't see, right?
no.
Okay, just wait a second. I'll stop sharing and start share again. Just my full screen instead of that particular window. I think this should be fine. Now, do you see?
Yeah, now it
Okay.
is it's perfect.
All right. So I have about 10 minutes or 12 minutes, so I'll try to stick to the time.
12
Um,
minutes good enough, 13 minutes good enough.
okay. Thanks a lot for inviting me for this talk. So my lab works on at the confluence of um, genomics and artificial intelligence for the last 5 years. And the genesis of my research was in Indian Statistical Institute, where Professor Pal belongs. And then I started my lab, moved to Delhi, the capital of India, and currently I'm also an adjunct professor in Australia, and I'll soon be moving to Norway, and will be closer to some of the speakers. All right, so today's talk is about how we can treat cancer, how we can predict what is going to happen after application of a drug just by looking at the gene expression data a priori. Okay, so the problem is very simple, that you can always apply certain drugs, targeted drugs and all that, but can you predict ahead of time that what is going to be the consequence, whether or not the drug is going to work, right? And that's where we can use AI, and I'll try to demonstrate with some lab data and all. It is a very, very recent project, and therefore it is not in publication. It is there is a bioarchive archival of this manuscript, which you can check out later on if you wish. Okay, so there's a myriad of okay, I don't think I have activated my video, and I'm sorry for that. So I think there was a request and I ignored, and now I have activated, although I don't know whether it will look very well. Okay, anyway, so I think it's not visible. Just give me 1 second. I'll move to a slightly more visible uh, location of my room. Um,
Now
yeah.
it is okay. Now it is okay.
Yeah, sorry for that. Very unprofessional of me. Now, you see, what has happened is over the last few years, tens of $billions have been invested to come up with drugs, right, that target certain molecules which are present, because cancer cells are our body's own cells, right? And it is the most difficult part here is the fact that cancer is extremely heterogeneous. And the second thing is, how do you recognize certain cells to be different in your body, and then you attack those cells? Your attack has to be directed towards the cancer cells alone. And cancer happens to have a lot of hallmarks, which are described here, such as angiogenesis, which is formation of blood vessel to supply more resources to the cancer and all that, activation of invasion and metastasis. So everything that is relatable to cancer cells as a differentiator from normal ones can be in principle targeted. And the science has moved a lot forward in the last 10 years, 20 years, and we have a myriad of drugs targeting every aspect of what makes cancer different from the norm. But the problem is, cancer is smart enough to dodge these attacks. As a consequence, the overall survival benefit that you accrue by application of any of these drugs is very, very limited, right? So if you look at some of this mind-boggling stats, you'll realize that only less than 10% of patients can be coupled with targeted therapy. So when somebody has cancer, you go for diagnostics where you find out what are the targets in your cancer that can be potentially, um, you know, leveraged to deliver a drug. But it so turns out that the human genome is so gigantic, uh, even if you sequence the entire human race, you will not learn enough. It is a sparse matrix problem, right? You cannot run a recommendation engine successfully on that. So therefore, you find targets only in a very small number of cases. And I don't want to go through each and one of the statistics, but for example, in a randomized controlled trial, they discovered that the survival advantage was just
Debarka, you got muted.
Please unmute yourself.
I don't know how I got muted because I was not operating with my cue. I mean, maybe somebody muted me by, anyway. So can you hear me now?
Yes, yes.
So what I'm trying to impress you upon the fact that only in few cases you get to see targets, and the overall benefit of survival, because you want to live more, right? Once you apply a drug, you expect that the patient will live longer. And it so turns out in the randomized controlled trial that the survival benefit is just of 0.3 months. And you spend enough money, five to $8,000, just to find out that you have a certain target. But what is not told today to the cancer patients is that even after application of all these drugs, you cannot expect a better survivorship, right? So they go, you say, I found target, I'll apply this therapy, but you don't tell them that even after application, I'm not sure whether the cancer cells will become rogue and they will resist further therapies. So this is a bit of a biology which essentially says that when you apply a target therapy, some of the cells indeed die, and you may see a quick response that some cells are dying and the tumor is shrinking, but then genetic resistance develops, right? And of course, genetic resistance with contribution from the tumor microenvironment specific factors and further epigenetic reprogramming, you end up creating a lot of heterogeneous pool of cancer cells that are drug resistant, and they proliferate very fast, and eventually they metastasize and the person dies. So the question that I wanted to solve, and we wanted to solve as a lab, is how can we make a prediction method for telling the patient that, okay, this is the chance that you will develop a resistance to the targeted therapy that you have been given with, right? And there are so many different ways it can be achieved, that there are several readouts for your genomic data. It could be DNA mutations, copy numbers, gene expression, DNA methylation, microRNA, proteins, and of course, clinical data. And pretty much today, all these methods are being used heavily to see that if we can predict what is going to happen. But we chose to use gene expression because it is well understood, it is cheap. There are a lot of public resources which you can use for artificial intelligence based approaches, building models, training new ensemble classifiers and stuff like that. So we chose to use gene expression, right? And I'll try to explain how it works. So on the top, you see that we gathered a lot of data from the public repositories of cancer cell lines. Cancer cell lines are immortalized cancer cells, which you can use in your lab to apply drugs and see what happens. So we got gene expression matrix of the cancer cell lines, and these are untreated, right? So these cells are not treated. So this matrix has genes on the rows and the cell lines on the columns, and you have numerical values in this matrix which are marked by the different colors, and you have patients, and from patients also from consortia, TCGA kind of consortia will give you a lot of patients and their gene expression of treatment naive patients. They are not treated yet, because if you treat, their gene expression will undergo changes. So these are before treatment, and you get these matrices. You can combine these matrices using pathway projections, which use some kind of random walk based methods to find out which pathways are enriched in a particular sample. And that also mitigates a lot of batch effect. In statistical genomics and machine learning, batch effect is a big problem that we need to deal with in order to make something useful, right? And of course, what we also get from the right hand side, as you can see, is what drug is applied and what is the signature of the drug, because drug is a molecule. A molecule can be numerically represented as a signature or a bunch of numerical values that explain what drug it is, right? The structural elements of the drug can be presented as a vector. So you get a gene expression vector, you get a drug vector, and you get response information. So your decision variable is response, and your explanatory variables are gene expression data and the drug descriptors. And this is how you consider you construct a supervisory learning problem. You can solve it using a classification method. We used a particular ensemble of different classifiers to predict drug response from gene expression data. And it is pretty accurate. There are so many nitty-gritties I cannot just describe here, but it is not so straightforward. It so turns out that merging these two different kinds of data is also very difficult. And so there will be batch effects. One data gives you IC50 value for the in vitro experiments, and other data gives you clinical responses. So merging them itself is a very big task. So here you see how predictive our models are. On the left-hand side, we show a validation on bulk RNA sequencing data or bulk gene expression data of different cancer cell lines. On the right-hand side, we show the same thing for single cell data. They are slightly different because a single cell, you can look at, at a very high resolution into your tissue architecture, particular clones of cancer cells. So this is much more powerful and has been largely unexplored. So we collaborated with Queensland University of Technology, where I'm an adjunct professor. So there is a group of Colin Nelson, who is a prostate cancer research veteran, and they do a lot of animal experiments. And they did once for certain experiments for our model, and it was quite a pleasant surprise for us that with how accurately we can predict what is going to happen in mice, right? So we used animal models here. So if you see the left hand side, so what happens is the tumor keeps growing, right? Once there's a tumor, it keeps growing. Then and this is prostate cancer, right? These are mice which have been injected with cancer cells to convert them into, to mimic what happens in patients, right? So they grow tumors, and when the tumor grows, the first thing you do is castration. Then castration actually reduces the burden for some time, and then it again spikes. So this phase is post-castration. Then this is castration resistance. So castration resistance is cropping up here. And then at this point, to apply a novel drug, enzalutamide, and then it goes down, and then they again become resistant to the drug. So this is how cyclically the tumor evolves with exposure to drug and castration. And by the way, enzalutamide is a molecule which was not part of our training data, but by the virtue of the construction of this problem, we represent drugs as vectors, so we can predict the drug response for vectors, drug response as we predict, the response to a new query in a machine learning system, just because I can represent any drug as a bunch of numerical values, right? So it can also be used for finding lead molecules in drug discovery process. So these are the different categories, like, pre-castration, post-castration, castration-resistant ones, enzalutamide-treated ones. So we can accurately identify that with enzalutamide, application of enzalutamide on these tumors, you are going to give rise to resistance, right? So these are all predicted values, in terms of z-scores. So this is a cell line-based model. So we have written IC50. The lower the IC50, the more sensitive the drug is to the tumor, right? So we could correctly identify that enzalutamide, these mice are going to be enzalutamide resistant. And this is a cluster view in the UMAP embedding space of the samples. Not much required here to understand what is going on. But we could also cross-validate this with the proliferative index of all these different subtypes. I didn't discuss about the architecture, the neural network architecture of the model, because I don't think we have the time and space for accommodating that. But we also tried to see what happens in TCGA, because TCGA presents you with a lot of um, patient tumor data, right? So where you have the response, like you take the treatment naive cancer cells, you subject them to RNA sequencing, you get the gene expression readouts, and you know what happened to the patient when he's applied with a certain drug. And you try to model that using similar kind of methods that I just described, the similar kind of architecture and framework that I just discussed. And with that, you see the red ones are patients which did not follow our advice. That means for them, we predicted the applied drug to be wrong, right? Will not, uh, the patient will not respond to the drug. And the blue ones are ones in which case we said, okay, they are going to work. And you see, there is a massive difference. And this is quite stark to us. And we were very happy to see this result. And if you translate this into a human understandable language, this means that with use of AI, we can get a 70% increase in the 5-year survival of the patients. 70% increase in the patient number for 5-year survival. 5-year survival in cancer on an average is a very good thing. It's considered as good. So in a sense, what we learned is simple RNA sequencing experiment, which can be done in many clever ways, and the cost could be even less than 10,000 rupees if you use 3' technologies and all that. So just using that, you can predict what is going to happen when the drug is applied. We tested that on animal models and bioinformatically on patient data as well, because on patients we cannot do a clinical trial like that, right? Because it still needs to be proved. Prostate cancer is a very elusive cancer, and its biology is very complicated. And I do not. It is less. It is not as well understood as breast cancer. Let me put it in that way. Okay, there are less known molecular subtypes of prostate cancers, and these are the cases where uh, such machine learning based approaches can be really helpful on the ground. Okay. And the other byproduct of this approach is the fact that we can predict treatment response for drugs which my model has not seen before. This is also a great advantage, because all of you might know that it takes roughly $10 billion on an average to create a
Uh,
drug.
Professor Debarko, please wrap up.
Yes, I'm done. So this is the paper which from where all these results are shown. Please feel free to go into bioRxiv and check that out. And I must mention that Colin's lab contributed a lot in terms of the animal experiments and in vitro ones. And this is my collaborator network all over the world, including Europe and Nantes. I think there's a presenter from Nantes, and Professor Thierry, who is one of the leading EMT researchers, and the Indian counterpart, and Stephanie is a breast cancer key opinion leader, Colleen, and so on and so forth. With that, I really wanted to thank you again. I'm not sure because I turned my back to the clock, so now I don't know how much time we have taken additionally. Thank you so much.
Thank you very much for an excellent presentation. So maybe you may please now stop sharing and
Yes, I'm trying to do so. I think I have stopped sharing already.
Yes, yes, yes.
I don't know why I can't stop my video, although I have stopped my sharing. Uh, Professor Mukherjee, can you use your sharing?
Yes, yes, I'm
Okay,
just.
okay, great.
Can you see my screen?
Yeah, we can.
Okay. First of all, thank you for this opportunity and sorry for the mess up. I come from IIT Jodhpur. I have recently actually moved to IIT Jodhpur. I was earlier a chief scientist at the Institute of Genomics and Integrative Biology, and done 23 years of research in genomics, and now I moved to an engineering institute. So today I'm going to talk about some initiatives which I have sort of proposed over here with a transdisciplinary outlook. So this is a group of people who would be working in this area, and I'm a very different area from what all you are. So this is like a domain which needs a lot of AI and data science tools. So maybe I will be able to kind of project that aspect. So going to the next slide. Now every one of us appreciates that the global burden of disease is immense, especially in the area of non-communicable diseases, because as the longevity has increased, the health longevity has reduced nearly by 10 years, and there is now actually enhanced need for the more scalable, personalized, and preventive approaches so that we reduce the health burden by preventing those 10 years early onset of the non-communicable diseases. Now, we feel that if we want to move towards that, there is a need to have a paradigm shift from our traditional approach of how we discover treatments, because they were all mostly population based, and that has highlighted that how population based studies are really imprecise when it comes to individualized outcomes. And now the time has come for a move to a one person kind of trials where an individual becomes his own control, and we need newer and newer approaches to be able to do that. Now, there also what is getting really appreciated now with all the genome sequences, microbiome sequences, and also the context of epigenetics molded by various factors, that how we define health and healthy is really a challenge. We don't have a definition of health and healthy as yet, and unless the idea is that people are trying to do various kinds of methods, and this is where now people are moving towards how you can define an individual based on the exposome, the epigenome, the microbiome, all the kinds of things which Debarka was talking in the context of cancer also, to identify whether there are pathways or networks which are different, and because of which you can actually evolve an individualized treatment. So this is basically being built by capturing a huge amount of data and then trying to use different kinds of analytics to identify such kind of patterns. Now, such kind of medicine of personalized approach is also not new to India. So this is now being recently acknowledged, but in India, there's a system of medicine called Ayurveda, which also has approaches which tends to cater to maintaining health in healthy and alleviation of disorders and disease. These are Sanskrit shloka dating back to 5,000 years, but there is a definitive element of precision medicine described, and it is in practice today. And what we've been doing for the last 20 years is trying to develop methods where we could kind of interrogate this principle, which has stood the test of time, and see whether we can actually arrive at a better way of stratifying people. And this is what is we developed the area in the country called live genomics. And the idea is that and especially in the context of India, where there are huge number of communities, endogamous groups, identifying individuals who would need a personalized treatment, we thought that if we can actually have a stratification approach where we are able to identify differences amongst the healthy, then would we be able to reduce the risk of or have a much better outcome of disease. That is what is being defined by number needed to treat or number needed to harm. So if you are able to reduce that to, let us say, by stratifying, you don't treat these people and only you treat them, then you harm also lesser number of people. So why Ayurveda we think is very powerful is because Ayurveda has a systemic definition of classifying people on the basis of multi-system attributes. And based on that, it's a scale-free measure where individuals are classified onto 7 broad constitution types, and that predicts their health response, disease trajectory, and how you manage the diseases. So I am cutting a long story short. We have been able to develop over the last 20 years a way to utilize this information of stratification. We've discovered genes, for example, related to hypoxia based on classification by Prakriti. And based on those genes, we've been able to see whether they could be targeted in diseases which are relevant in hypoxia. And then we try to identify whether we would be able to discover more such connections or networks because of the multi-system attributes of the constitution types. And if we discover them, then would we be able to evolve a therapy which are contextualized, let us say, in asthma from the hypoxia axis. And whether the Ayurveda medicines which are used in such conditions actually go and integrate to this very axis, then that closes the loop of the understanding of the network medicine. So we build these kinds of frameworks. And also additionally, we have built frameworks based on connectivity map to interrogate multi-component formulations of Ayurveda for probing what is the polypharmacological basis of such drugs through the connectivity map. And based on this, we are also trying to give a molecular explanation of the basis of Ayurveda preparation. So this is one recent ones which we published in scientific reports, which where we looked identified that there is one such formulation which is used to treat hormonal disorder and fever, links ESR to viral inhibition. So having developed those frameworks at IIT, we actually aim to develop a transdisciplinary center for precision, meaning excellence for integrative precision medicine, where we would be able to, since we are getting a molecular interpretation of Ayurveda, would we be able to link it with modern medicine? But that requires a lot of interdisciplinary effort, building ontological links, etc. So IIT J has a school called School of Artificial Intelligence and Data Science. So I'm an affiliate faculty that allows me to integrate or to interact with a large number of fellow colleagues from different engineering sciences. And that's how we've developed this transdisciplinary framework, where we try to see whether we would be able to have a phenomics based way of non-invasive stratification as is described in Ayurveda, by integrating these principles with different knowledge systems and integrating tech solutions for objectivity to phenotyping, and then how we apply it to management of rare and common diseases and develop an integrative framework. But what we want to also do is we want to test it out in a specific arid region. The IIT has lots of expertise in all these aspects, not me, but my colleagues from different departments on smart healthcare, IoTs, integrating health records, multi-omic analysis we are building up, and AI ML is a forte of the computer sciences department. And we also have interdisciplinary research platform where also again the investigators from different electrical engineering, mechanical engineering, computational sciences are trying to develop smart healthcare devices and sensors and solutions. So this is what we would aim to use. And then I explained why arid, because we are in a very we are located in a desert region, which is the most largest and populated desert in Asia. It also is divided into dry Thar and wet Thar based on the Aravalis. And there is an extreme heat, aridity, scarcity of vegetation, all those which will drive an individual to their ultimate physiological ebb. And also there is a lot of scope for bioprospecting because it's also very rich in minerals. So we thought that if we would be able to focus our study by integrating the ecosystem of Thar, then we might also be able to evolve precision studies in the arid ecosystem. And that will also be very useful and pertinent in the engineering institutes, because deserts have these designs which allow them to adapt by maybe being evaporator phenotype, evader phenotype. So there are different kinds of adaptations because of which the animals are able to survive in such extreme conditions. And Thar Desert also has many such animals, endemic animals. And that's why we kind of evolved this Thar designs, which is desert ecosystem innovations guided by nature and selection. That's because all the deserts are formed in different time zones, but because of the extreme environment, they also provide natural laboratories for biological designs, because there are these environmental triggers which would kind of lead to adaptability, adaptation, or selection for an individual to thrive. And many of them, you see them as recurrent phenotypes, which could be at all these kinds of levels. And when people have studied various deserts, they have found that there is convergent evolution, which allows us to identify the design principles which could be of utility to all the different sciences. And also desert have traditional cultural systems which allow them to conserve. So we have to contextualize any precision health in the context of the culture. So we thought that this is an interesting test bed to do the precision study. And this is pertinent because there are many studies around the world where they have actually shown that if you have desert rats and you take them to the laboratory condition, this is like a rat from Ethiopia. And 1963, these experiments were done. You take them to Ethiopia, they develop all the things about cataract, high blood sugar, and all within one generation. You do a second round of experiment where you actually feed it healthy, only fresh vegetables, and they do not develop such phenotypes. It's been shown that these desert sand rats actually have mutations in genes that allow it to adapt to high aridity and low calorie intake, and any movement to a non-native environment makes them such obese. And now this kind of a thing is also being used in phylogenomics approach, where people are looking at different animals, different behaviors, and trying to see whether they could fish out genes related to things like mammalian obesity. There's a very recent paper from such kind of extreme phenotypic adaptation. And that's where we thought that the desert design will also come in very useful in the medical context here. So the idea in the Thar designs is if we can actually study in an integrative manner the animal, the environment, and the human health, we might be able to identify different signatures of ecosystem, which so this is where the big data comes in. So we actually have an umbrella of a transdisciplinary network where we have kind of got all the people in one platform in the Jodhpur cluster. And here is where we want to study the ecosystem phenomes from macro to micro level, identify patterns using computer vision and ML with domain knowledge of various areas, and develop predictive models. So that's what is the basic idea. We capture all kinds of phenotypes and then use big data architecture to integrate them and identify patterns, and then finally develop a toolkit for recommendation engines. So the idea is that if we identify such signatures, we might be able to know what are the specific intervention needs of the ecosystem. Plus, there are other things of desert medicine, which is also described in Ayurveda. So we can integrate the frameworks for discovery as I described for network medicine. And that's what we aim to develop. And we also will integrate the Ayur tech solution for integrating Ayurveda to future proactive care. And the aid school is allowing us to develop that. Ministry of Ayush has shown an interest as to how we can maybe use a lot of data analytics instrumentations to be able to understand the principles and practices of Ayurveda and integrate it in modern medicine settings. So I just wanted to give a flavor of what we are doing, and we would be very open for collaborations of different kinds. As you can see, it's a broad canvas, and we are just starting up. Thank you.
Thank you. Thank you very much for respecting the timing and for these very valuable talks. And it was very interesting. So thank you very much for this. So now it starts to be the Q&A session from the first Q&A session of this morning, or afternoon for India, sorry. The idea here, just before, we will ask you to, if you don't mind, to open your camera just to take pictures of a souvenir of this morning session, if you agree with. So if all the people can open his camera. Oh, unfortunately, we have one speaker that has just left. Maybe there is a problem. And so I'm sorry, but because the co-animators has a problem of electricity at home, so it has been disconnected. So is there for the other people, can you just open your camera, either for the attendees, if you agree?
Hi, this is Vivek here from Tech Support. I'm requesting you please switch on your camera. So can I take a screenshot? Is it done?
Okay, I think so. Please proceed.
Yeah, it's done, sir. Thank you very much. Thank you.
Thank you. Thank you very much. So now I'll start the real Q&A session. So do not hesitate to unmute your microphone in order to ask questions to our three speakers. Please.
Yeah, may I ask a question to Jan?
Yeah, sure, with pleasure. Go on.
Yeah, Jan, you were basically talking about using the microbiota data to do characterization of patients. So there was a big paper published in Cell this year from the Metasub Consortium, where they are showing that, I mean, they are basically collecting microbiome data from different corners of the world. And they're saying that depending upon the temperature, humidity, and many other factors, the nature of the microbiome basically changes. So do you feel like this work can actually be extended to the other patients from other countries? You know, like, do you expect that the microbiome profile will be different in the patients from different countries, first of all? And then what is the difference between the microbiome profile of the patients and the normal subjects?
Okay, so it's a very, very good point. So definitely the microbiota is influenced by the disease, of course, but as you say, by the environment, by the diet changes, by many, many other things. What my feeling, okay, so first, on the data we've been playing with, we got rid of this external variability because we have a cohort of people that are all in the same region. So at the moment, we're getting to the easy part, let's say. But then on the broader task, my feeling is that, but this has to be confirmed, my feeling is that even though the microbiome can be very different in terms of the name of species, if you reach out to external knowledge and function and metabolism, maybe you find something that is much more robust to external changes. You know, you might not have the same species, but they do the same function in a way. And by having other representations of the patients, maybe you are more robust to environmental changes, diet changes. But this is just a speculation. But this is a hope. And the second question, I completely agree with you that in terms of the question was about what is a normal microbiome and do you want to you want to compare patients to the normal microbiome. And what's interesting in terms of Crohn's disease, most of the paper, this is what they do. They have a control group and they have a patients group, and they compare the two microbiomes. And I agree with you, I'm not super convinced with these kind of approaches. And this is why what we did is just to take the Crohn patients and try to do unsupervised clustering on only those patients, and not to look for external references. And I think there is much to be gained with these unsupervised or self-supervised approaches just on the patients with a given disease, because the reference is super hard to find. And again, if you have a control group from your region or other region of the world, would it be the same? It's not sure at all. And the last part, although I do agree that the big projects of having a lot of microbiomes for different corners of the world are super interesting, but to me they are more interesting in the sense of training large unsupervised model to find the structure in the microbiomes. And then from this model, what I hope one day is to fine tune on your specific data set. You know, this is more the kind of hope I have, rather than take off the shelf and make it work right away.
I appreciate your views. Thank you.
Thank you very much for the question.
Thank you. Do you have other questions? I have a question. It's a traditional question that I ask to every speaker since yesterday. The idea, I will start with Professor Mitali Mukherjee, if you don't mind. Imagine that you have full funding, infinite funding. What should for you be the most important and the most urgent research topic that should be investigated in the next 5 years in the AI for healthcare?
Can you just repeat the last part? You are saying, imagine if I am fully funded, what is the most pressing problem I would like to address? Is it?
Exactly. In the AI for healthcare problem specifically.
So for me, I think I now feel that we need a very precise definition of how you will define health in different environmental conditions. So if whether the definition of health by molecular parameters would be same for natives versus non-natives, because that would determine maybe the baselines of what you call as intermediate pathophenotype, and which actually is the interventionable part. So from our studies on when we stratify healthy people, we observe that healthy people have differences in baseline phenotypes of those very processes which are kind of like cell proliferation, inflammatory response. So if the baselines of those vary between the people, then the trajectory to the disease or the treatment needs or the basic network modules of them would vary. So we need to understand the health baseline of an individual to be able to calibrate the disease very precisely. So that is why you need to contextualize it, and unless we do that, we are still working with population averages, and most of the time our studies are moving from disease to getting them to health. But I think health to health transitions and to disease, we need to have definitive markers and endpoints. Thank you.
Thank you very much. Jan, same question for you, if you agree.
Yeah, very last question. Okay, maybe use this mic like this, it's going to be better. Yeah, to me, the key question is all about basically building AI on top of what we already know, and not just on top of the data set that you have on your hands. Because you have had many papers over the past few years being like, basically, I don't know anything about radiology, and look, I'm beating radiologists. And I think we have to go beyond that and try to really mix experts from different fields and the AI algorithm and make them work together for better performance and better interpretability. And this will hopefully help broaden the application of AI to health, but to any kind of domains where people are interested in this. And so there is building on expert knowledge plus AI, and then having the training programs to also, you know, bridge the gap between people training to AI, hardcore AI, and people training to a discipline, and bridge this gap so that communities can discuss a bit easier. That would be that.
Thank you. Thank you very much. If the attendees have other questions for our speakers?
Yeah, I have one question. Am I audible? Yeah, am I audible?
Proceed.
Yes, you are audible.
Okay, so yeah, this is Dr. Joshi from NMIMS Sherwood campus. I'm having one question particularly related to the conjunctive works of AI as well as the chemoinformatics. So, like, can you anyone suggest something about it?
Sorry, I didn't get the question correctly about AI and chemoinformatics.
Yeah, I repeat, it is artificial intelligence with chemoinformatics, like the decision support system in which the chemical-based compound can be identified using AI algorithm.
I think it's had a lot to do with Debarka presentation, I guess, who's not around anymore.
Yeah, unfortunately, I think he has a problem of connection. Debarka just disappeared at the end of the Mitalis presentation. And
should I write my query into the chat box?
Sorry, Dr. Joshi?
Should I write my query into the chat box?
Yeah, maybe. So if he's able to come back for the discussion, we can send it to him again. Thank you. So in the meanwhile, there is any other question for our speakers? If it's not the case, I would ask Dr. Ramakrishnan, Subhasri Ramakrishnan, to take the floor to share their slides and to prepare for the next presentation. So Professor Ramakrishnan is an additional professor at the Department of Neurology of the National Institute of Mental Health and Neuroscience. And we are very glad to have her this morning for this session. And so she will talk about brain-computer interface in neuro rehabilitation. So please proceed. The floor is yours.
Thank you for the opportunity. Very good afternoon to everybody. I'm Dr. Subhasri. I'm additional professor in the Department of Neurology in MHANS. And so basically, I'm a neurologist who deal with patients with neurological disorders like stroke, cognitive disorders, and we get to diagnose, manage, treat, counsel them. And my research interests include cognitive disorders, stroke, and also brain-computer interface. So I have a fellowship, Fulbright Fellowship at the University of Houston, where I was lucky to have an opportunity to work with Professor Jose Contreras-Vidal on non-invasive EEG-based brain-computer interface. So today I'll just briefly introduce about BCI and its clinical applications and its role in neurorehabilitation, especially with reference to stroke. So brain-computer interface is a neuroprosthesis which interface with the central or the peripheral nervous system to restore the lost motor or sensory capabilities. So basically, it's a communication which is based upon the signal activity that is generated in the brain, and it works detouring or independent of the normal muscle and nerve pathway. So basically, when the brain thinks of action, of moving a hand, for example, it is through several organs of several different structures of the brain, and then through the muscle and the nerve. So what BCI definitely does is that it bypasses this circuit and causes action. And this is a non-muscular communication and control channel. So there are various different types, invasive, semi-invasive, non-invasive, based upon the penetration of the skull. And there are exogenous and endogenous BCI, which is based upon the brain signal, whether the signal is external, the stimuli is external, or whether it is internally arising from the brain per se. And these are the components of brain-computer interface: signal acquisition, feature extraction, translation, and device output. So the BCI signals, the non-invasive and the invasive ones. So these can be procured through different parameters like the EEG, the electroencephalography, the FNIRS, the fMRI, EOG, MEG. So these are the different techniques by which the brain signals can be acquired. And some of the important signals, these include the EEG signals, the evoke potential signals, the event-related desynchronization potentials. So these are some of the signals which are procured from the brain. And these signals can be classified based upon whether it is electrical, whether it is magnetic, or whether it is metabolic. And then these signals are processed, and these encode the user's intent or the message or the command. And there are various features that can be looked into, and various spatial filtering techniques that can be looked into. And these are the various algorithms which are commonly used in BCI, which includes LDA, the support vector machine, and neural networks. So these are some of the algorithms which are commonly used in BCI. So basically, signals are acquired from the brain through various techniques like EEG, ECOG, and fMRI, and then they are processed and then translated. The user's intent or the command is read, and then it is sent to the effector device, which can be a prosthesis, it can be a wheelchair, or it can be a cursor control or any assistive device. And then this closes the whole loop. So this is the whole overall idea of brain-computer interface. And what is the clinical application? So to a clinical neurologist, how are they going to help? So these could be assistive, where it can help to restore a lost function, or it could be rehabilitative, where it helps to foster neuroplasticity. And for example, as in the case of stroke. So stroke, when a person has difficulty, focal deficit, that is, say, for example, they have motor weakness of the upper limb and the lower limb, they have difficulty in swallowing, they have difficulty in communicating, they have cognitive deficits. So there are a lot of domains which are affected by the neurological disorder, and BCI has a major role in clinical applications. Not only stroke, there are a number of neurological conditions like the amyotrophic lateral sclerosis, the brain or the spinal cord injury, cerebral palsy, muscular dystrophies, dementia, movement disorders, locked-in syndrome, disorders of consciousness. So there are a lot of neurological conditions, which some of them have treatment and some of them may not have a symptomatic cure. And BCI has a role in them. For me, EEG is one I'm interested specifically with EEG-based brain activity measurement. And there are different types, the dry ones, the wet electrodes. There are different headsets and different modalities of acquiring the EEG, the brain signals. And there are different control signals which can be studied in them. For example, the sensory motor rhythm. So I would like to focus on the sensory motor rhythm. So when someone thinks of moving a hand, so this causes a small drift in the EEG signal, and these can be correlated by the EEG. And these are, say, for example, the motor related cortical potential, the event related potentials. So these can be picked up from the EEG when somebody wants to move their hand. And based on that, the movement intention can be detected. And this has very good implication in rehabilitation. So for example, this is called a new rhythm. So what happens is when someone thinks of a movement, or when someone prepares for a movement, there is a dip in the rhythm contralateral to the side of the movement. So these are called as the sensory motor rhythms. These, over several training sessions, can be helped to control a cursor in a computer, to spell a device, or in an assistive device, or in a functional electrical stimulation, prosthesis, or wheelchair. So there are very many applications to this. So this is what I was mentioning. So when someone is moving a hand, for example, or even imagining a movement, so that causes a low frequency dip in the contralateral hemisphere, which is called as the event-related desynchronization. And over several training sessions, these require long training sessions, the person will be able to control or train their sensory motor rhythm. Thereby, that can be coupled up for clinical applications or for the rehabilitation of the person. This was one of the initial papers where cursors, it's a simple one-dimensional cursor control, which is done by sensory motor rhythm modulation. This is the event-related desynchronization, where there is a dip in the potential, which can be recorded by the EEG signals. And these can be used in chronic stroke rehabilitation to, as I mentioned, for controlling the orthosis, or it can be used to control a finger movement. And so that is movement. Then there is another component in the EEG, which is the motor imagery based BCI. So what is motor imagery based BCI is this: the person is imagining a movement without actually moving the body part. So what is interesting is imagination of movement more or less activates the same brain areas which actually causes the movement or motor observation. So same brain areas are activated, and this again can be used to trigger neuroplasticity and activation of the brain areas which are involved in planning and execution of the voluntary movements. And there are several papers on BCI and EEG based and motor imagery based BCI and stroke rehabilitation. And the studies have definitely shown that in comparison with standard rehabilitation program, combining BCI with the standard physiotherapy definitely helps in the post-stroke rehabilitation. So this is, I hope the video is visible. So this is one of the study as a part of my Fulbright fellowship, where this is a subject, the consent is taken from the subject. The subject has upper limb weakness due to stroke. And here you can see that she is imagining movement. So basically she imagines that she is opening and closing her hands, and that causes sensory motor rhythm modulation. And over several training sessions, she will learn by modulating her sensory motor rhythm to control the cursor or the ball on the screen to hit the target. So she's not moving her hand, whereas she is imagining movements, and that is acquired by the EEG, the sensory motor rhythm modulation, and then that is triggering the movement of the ball. This is another video which I wanted to show. So this again, we had carried out at NIMHANS in collaboration with IIT Palakkad. So here what we have done is, this again is a subject. This is a subject with a stroke where he had right upper limb weakness. So here there is a visual clue that is presented. The person has to imagine that he is going to move his hand towards the side of the visual clue. And that motor imagery modulation, the sensory motor modulation, controls the robotic arm, which is attached to the right upper limb, and which in turn controls the robotic arm to go towards the direction of his imagination. So this direction decoding, we had a classification accuracy of around 74% in around 16 stroke subjects. So you can see the video here. So there's a visual clue. Patient is imagining movement of his right hand. That triggers the movement of the robotic arm, which is attached to the stroke affected hand. So in this paper, we were able to decode the direction of the imagined hand movements in stroke subjects. And this we feel is will be a good advancement towards the rehabilitation of stroke subjects. And this apart, BCI with EEG can be also used to control ankle foot orthosis. It can be used along with the functional electrical stimulation to control and grasp prosthesis. It can be combined with exoskeletons. It can be used to control wheelchair. It can be combined with orthosis, prosthesis, assistive robots, and in NeuroRex project, which was carried out at University of Houston, my mentor over there. So they had also showed that EEG, the BCI based ones, were able to control the movements of the subject based on the motor imagery per se. He was able to walk with the powered exoskeleton. And so that was about motor imagery based BCIs. Then there are other signals, say for example, the P300 spellers. These are assistive devices which has utility in post-stroke aphasia. So the person thinks of a word, and based upon his focus on a particular word, based upon the signal that is detected from the brain, the 300 milliseconds after the event is seen or heard, and that can be used to predict the words. So this is assistive BCI. And then there are vibrotactile P300 based BCI, which is useful in patients with disorders of consciousness, where they want to communicate to the others by saying a simple yes or no. So in communication, there is a application. Then there are steady state visual evoke potential, where again, when person is not able to move, but he wants to communicate to the external world, just a movement of the eyes can be coupled up and can be used to express the binary expression intention. Then there are several other hybrid BCIs where EEGs are combined with FNIRS or SSVEP with EEG. So there are hybrid BCI, and in the market there are techniques available based on this hybrid BCI as well. And they have also shown, say for example, in Recoverix, motor imagery with functional electrical stimulation and VR technology, people have shown that it has a good advanced improvement in those suffering from stroke. So there are several techniques of procuring the EEG signals. There are different types: metabolic, electrical, magnetic, and these are mostly non-invasive, portable ones. And these can be coupled for assistive or rehabilitative BCI. And I am specifically interested with the EEG based or the P300 based BCI, and we have some experience in working in this interdisciplinary area, where we have combined with other IITs, and we have tried to answer some of the questions of this interdisciplinary area of BCI. There are still a lot of difficulties, practical difficulties, and difficulty in acquisition, validation, and it is still an evolving field. A lot of multidisciplinary team and further clinical trials are required. And there has to be a bridge between the engineers and the doctors. So this is still evolving field, and but it will definitely be a very potential and an advanced treatment option, and it will assure a new era of human creativity, and it can be brought about by intimate fusion of brains, machines, and computer technology. So I'm interested in the rehabilitation of patients with neurological disorders, especially stroke and cognitive disorders and other neuromuscular disorders. And I'm open to collaboration where healthcare can improve with the use of interdisciplinary, collaborative, and research options. And thank you again for the opportunity.
Thank you very much for your valuable talk. So Nikhil Pal has just come back, so I hope he will be able to take the lead for the Q&A question for the next part. But just before that, we have a second talk in a row. This second talk in a row will be given by Akash Narayana, which is senior data scientist from the Narama Health Analytics. So please proceed. The floor is yours. It's perfect. We see your slides. Yet it does not move, but I think it will do. Okay, perfect. Please unmute your microphone. You are muted actually.
Yeah.
Yeah.
Hi, everyone.
Yeah, we can hear you.
Akash, before I start, let me just check if I can see the screen. My slides move.
Yeah, slides move.
Okay, great. Yeah, thanks again for this opportunity. It's been a wonderful set of talks that I've been listening to. I represent Niramai, a startup based in Bangalore, India. And we have been looking at breast cancer screening with thermal imaging and, of course, AI. So this has been an interesting journey, and the motivation stems from the fact that in women, breast cancer is one of the leading causes. And in a country like India, we see that the detection of breast cancer happens quite late, and as a result, the survival rate is quite low, or the mortality rate is quite high. You see the numbers here. The numbers, are 2018, 2-3 years back. Now I think they're kind of around 2 million diagnosed in India, and close to 50% of them kind of die, unfortunately, because of pretty late diagnosis. And all of us are aware that breast cancer is something that can be easily treated if detected early. And that's the reason Niramai has come with its product called Thermal Analytics, which can aid in identification of breast cancer. So the motivation or the need for a different modality. Okay. So typically we have X-ray mammography, which is the gold standard for women. Please note above 40 years. And of course, ultrasound as an adjunct, and MRI for women with dense breasts. So what we see, you see in the table is, the cost of it, the ease of use, and the age of women and all that. This makes it difficult for women to screen early, especially in countries like India. And this is where we come with with a product which would make it more affordable and easy to screen for breast cancer. Note that when for women less than 40 years, they typically tend to have dense breasts, and X-ray mammography is not recommended there, and that's where you would probably have to go with MRIs and all that. These make the costs quite high. And of course, the privacy concerns are there too. And, and some of the setups have to be in, big towns and cities. So the reach is another important factor. So we have looked at thermography. So we looked at thermography that's essentially looking at temperature values on the breast surface. We know that cancer is more of cell proliferation due to angiogenesis. So, and this results in a lot of new blood vessel creation, that's angiogenesis. And there's a lot of heat produced as a result of it. So can we pick that up? Can we look at the heat patterns on the breast, and as a result of it, can we kind of say, hey, look, there's a potential tumor out here. There's a tumor out there. There's a lump out there. Can we pick these things up? Yeah. So the heat and so on. So we have good hardware which can do this infrared thermal imaging that has come up, so much so that they can capture variations up to 0.02 degrees. Yeah. So with this, good support in the hardware for imaging, we move on. And what we propose, so we have a product that has been in use. So what we have is, a scenario like this where the subject or the patient is in a very private setting in a room, alone with the camera, the thermal image camera, and a technician sitting outside, and the subject is disrobes, and based on the guidance from the technician would pose for the appropriate pictures. So this is non-contact, non-invasive. X-ray mammograms are quite a painful process because the breasts are kind of compressed and the images are taken. Whereas out here it is much more free. It's more like a picture being taken, and there's no radiation. It's just that we're capturing the temperature, the IR waves from the breast. Portable, lightweight. In fact, we've been using it in various clinics, hospitals, and even in shopping malls of late. So privacy aware people like this a lot, and it does work for women with dense breasts. It's a big a nice positive thing about breast thermography. Moving on, we would have an hardware device camera like this, which captures an image of the breast taken, and then on top of this, we have AI coming in, which would look at it, analyze the images and tell, hey, look, this seems to be tumor. Okay, so we really look not necessarily at as we say, just a clinical examination looking for lumps, but this kind of looks and finds where the potential cancer sites are. Okay, I'll move on. So these are the various images that are views that are kind of captured. So it's say, for example, there's a frontal view, as we call it. Then there's the side view, and there's a an oblique view and a side angle of the camera. So we want to capture the left and the right breast in its entirety from various angles. But essentially, these 5 views give us a very good representation of the surface temperatures of the breast. These are all combined together, and we have our image processing and AI techniques that run on this and help us decide, come up with a report. So taking these images, these multiple images, run your image processing, machine learning, deep learning, and all that, and we come up with a report which is given to the doctor. So let me, where is it from? Okay. So I wanted to show you the report. Sorry. Yeah. So a report, I believe you can still see the screen. So a report would be of this format. We have the details, names, and so on, the clinical details captured, the complaints, essentially. And these are the various scores that we're computing on, okay? Which would be, we would have kind of explained it to the doctor, what they represent, how to interpret them, and some sort of analysis of it, of the various features that we see. And then a very textual report is given for each of the breast, talking about what kind of thermal patterns are there, symmetry, asymmetry, temperature changes, and a clear conclusion talking about in a language, in oncology language, giving a BIRAT score and so on. Yeah, and this is better by a doctor too. So this is the output that we get out of the screening process. We call it thermalytics. And again, a question as to why the need for AI. So once you have a thermal image like this, in fact, a good doctor could just look at the images and try to interpret it, right? But again, subjectivity comes in. Sometimes the color map makes a big difference. You can, to the left, we're talking of the same subject out here, and to the left, the color maps are not properly fixed, and they miss out the changes in the left breast. Whereas with an appropriate color map, we do get the more of the variations in the temperature or the higher temperatures. A computer-based, image processing-based method would really help in picking up, enhancing the temperature variations, and our AI would help in really giving a clear verdict on it. So how do we go about the core innovation, as we call it? So essentially, we are looking at symmetry between the left and the right breast, the thermal distribution, the heat distributions in a breast, the vascularity, the blood vessel network. We capture it. We look at it quite closely. We try to understand the reach of it, the tortuosity of it, the diameter of it, the caliber, and so on, and look at hotspots or the outer spots that we see in a breast, the boundary, the shape characteristics. All these are captured. So various features, more than 150 features are kind of computed from each of these breasts, and they're kind of used to make fed into our AI models and to come with a classification. So the pipeline would be as follows. We kind of essentially we have the subject sitting in a room. and the technician outside, but as the image is taken, there's an auto check to make sure that image is properly focused. There's a cooling of the subject also done. We make sure that the cooling has taken place appropriately. And the tag of the image, is it a frontal view, side view, and so on. And then segment the images, this is the classic segmentation, the computer vision problems of you segment the breast, you segment the areolar or the nipple areas. We use deep learning algorithms for these two steps. And then we look at localized regions like the hotspot, the tumor areas, and capture the visual structures, the patterns, get the various features. And then we have, classifications like a random forest or a SVM to come up with a score to say, is it a benign or do we really see a malignant tumor? So we give probabilities out there, 0 to 1 kind of a number to say, hey, with what confidence can we say this is really a malignant tumor or not? Moving on, so a study like this requires a comparison, clinical effectiveness. So we are comparing our work with the mammogram, which is the gold standard. So out here, what we have captured is, so many times when we do a screening, the subject walks in without a symptom. So those are the asymptotic cases, but they might have a cancer growth at its very early stage. So those are asymptotic patients. So we see that our thermolytics is doing quite well. In fact, the sensitivity and specificity. So sensitivity is essentially capturing the positive cases. How good are we? And specificity are those negative cases or no cancer cases. How good are we in capturing them? And we see that thermolytics is quite equivalent to a mammogram, and in fact does better with on the sensitivity part slightly, with 92%, and with specificity too, it's equivalent. So this was a study done over 767 patients, and this has been published in the ASCO Breakthrough Journal. Yeah. Moving on, the other thing that I was mentioning earlier, so mammograms, these are a favorite of the doctors, but they have a challenge with dense breast. So we have two cases out here, the cancer. So we have to, on the top, we have mammogram, on the bottom, we have a thermal image, and the left and the right breast, both are kind of stitched together in the top. So we see that in the right side panel where we have cancer, the mammogram is not picking it up, whereas our thermolytics has picked it up, and with quite high sensitivity and with a good specificity to equivalent to a mammogram. So this has been a very good point for us, a USP for us for this particular product, that we can use it with dense breast, especially for women who are younger. Moving on, just another comparison, which is quite a obvious question. So, why do we need AI? Is this, if somebody is sitting with just a hardware camera, can they do a good job? So we kind of try to compare manual thermography with an AI-based thermolytics. We see that thermolytics is doing quite good compared to a human expert, especially with the specificity aspects. The sensitivity is similar, but whereas the specificity, we are better there with a human expert. We have the group as, so this work has been going on for the last few years at Niramai, a startup, and we've had several publications and a few clinical trials going on to take this work in a more firmer footing and show the reliability of it. We also have various regulatory clearances for the company. So we have the ISO and the other CDSU clearances from India, and we have the CE mark and MDSAP for thermalytics product. So we got the CE approval recently, so that now we can take this to Europe and other countries. Yeah, so I can come to the end of my talk. I'll leave you with this slide. So this has been a work where we are trying to reach out to a lot of hospitals, a lot of camps, camps of places where we can do screenings at a much affordable cost. So a lot of people, community comes in, and we also encourage private screening at home. So we're trying to reach out to a lot of women folk. We work closely with HCGA, one of the premier cancer hospitals in Bangalore, and also with Apollo Clinic, which is a chain of clinics all over India. So this has been a work where we're trying to reach out with our product thermalytics, ensuring that people are screened for breast cancer as soon as possible, and we have we kind of reduce the mortality rate, which is quite high. We can ensure that the mortality rate comes down. Yeah. So this is the work. We have planning some clinical trials in the rest of in other countries too, and perhaps that is where we would also need some support for collaborations going forward. Yeah. Thank you.
Nikhil, we are not able to hear you.
Okay, Ian, can you hear me now?
Yeah, it's perfect.
So let me first say two things. One is I beg a sincere apology for this power cut, which has not happened over the last 1 year at least. And this morning I sent you an email saying that we may have problem with internet, and if that happens, please take over. I didn't have any clue that we'll have power cut. So thank you, Ian, for taking care of the sessions in absence of me. Now, we have for question and answers, we have about 12 minutes. So I have not listened completely to the first talk by Professor Ramakrishnan. I joined towards the end, but the second talk I was present. So I open the floor for question, and I would request the participants as well as the speaker to raise questions, whatever they may have. We have enough time for questions, so. But since no one is asking question, maybe I can ask some simple question to Dr. Narayana. So the first question is, thermal imaging looks very interesting and very promising, but is there any downside when you compare with, for example, X-ray? I don't want to compare with MRI or even ultrasound because they are going to be more expensive in terms of capture analysis.
Yeah, we don't, that's a good question, So a typical doctor would go for X-ray, that's a mammogram, right? And that is, that's radiation. And typical breast cancer screening have to be done, very frequently, so that's not a good idea. As I showed in my talk, mammograms don't capture dense breast. These are women below 40 years. And we see good incidence of cancer in such women in India, especially in recent years. That is where we score, So we can take this, our device, our product, which can do a thermal image and works very well with dense breast.
You've also mentioned that when you use AI system, the performance is better with these thermal images. My question is, what is the scenario if it's an X-ray? And assume that the dense breast cases are not there, AI versus human expert. That is the reason for asking this question.
No, that's a good question. That depends on the domain. Okay, in the sense, an ultrasound is more difficult to pick. Maybe an X-ray. It depends on what are you looking at in an X-ray. So, so we're talking about let's say mammograms. You know, we are sitting with.
I'm talking about mammograms only, and thermals and screening of breast cancer only.
Yeah. So with mammograms, it is by and large the skill of the the doctor comes in. You know, you have to zoom in, look at. There's something called microcalcifications, calcium deposits, and so on. You know, which is so difficult to pick up, and so much of domain knowledge. You know, I mean, so we see that people go for second opinion, third opinion with such reports. So the subject, the subjectivity comes in there, and that's where I think probably an AI-based, validated AI-based method would be quite helpful.
No, my question was, if I compare the performance of a human expert on a mammogram, compared to, there are so many AI systems for for analyzing X-ray images, including mammograms. Like you have compared performance of human versus AI system, and AI system beats the human. But in case of X-ray, does a human being beats the AI system?
I'm sorry, I am not. I haven't really explored that space. You know, we have not looked at mammograms in so closely because we don't work in that space. Yeah,
okay.
So, yeah.
And for in your case, because it's I think it's a relatively new area, right? So in that case, I mean, how do you generate the domain knowledge here? Like you need to get information about whether somebody is having cancer or lump or microcalcification.
We work closely with cancer hospitals. We tie up with them. We have doctors who come in to talk to us. We sit with them. We understand what to look for, what to capture. You know, when we develop our AI systems, what to really look for. You know, what are the distinguishing factors and so on. You know? So there's a lot of discussions that go on before we really launch or deploy our models.
And their background knowledge is probably related to X-ray or MRI or ultrasound.
Oh yes, because that's what is currently being used. You know, so they're pretty good with mammograms, with ultrasounds, MRIs, and so on, and they are interested to see that okay, there's a new opportunity out here to screen a lot more women much cheaper, and you can take it out anywhere, and anybody can do this. You don't need an expert to go with this device. We do our camps.
Right.
So there are doctors who are ready to talk to us a lot and help us out in building up the product to a good.
And it's cheaper than mammogram. That's what you envisage.
Yes, very much.
Thank you very much.
So Dr. Narayan, am I audible?
Yeah.
So I have one question that since if we look from anatomical perspective, the particular technique is based on hypervascularity and increased blood flow resulting from the tumor associated angiogenesis, and that can be measured via heat, the temperature.
Correct.
So similar technique can we apply for skin cancer as well, since it is also the surface cancer?
Skin cancer, I'm not sure.
Anatomically, the things will be the same. Angiogenesis will be there, means the formation of new cells. So definitely there will be variation in the heat. So can we apply this fundamental for the detection of skin cancer? It is very close, because that's why this is striking into my mind.
Yeah, it's quite possible. It's quite possible. We have some research streams where we are looking at other cancers too. Yeah, we're looking at other cancers, other problems which are where thermal imaging can be applied.
Yes.
Yeah. This looks interesting here.
Yeah. Thank you. Thank you.
Thank you. Yeah, and I have a question for the previous speaker.
Sure, please proceed.
Yeah. So the question is, she was mentioning about P300. You know, I remember if it's a spell something. You know, spell completion or something like that.
Yeah, it is called P300 spellers.
Yeah.
So basically, it is based on the concept that after 300 milliseconds,
Correct.
after a visual or auditory stimuli, there is a signal that is evoked in the parietal region of the brain. That is P300. It can be visual or it could be auditory P300. So based upon the oddball paradigm, so when rows of numbers or letters are flashed in a computer system and the person wants to spell a particular word, so he would focus on one of the letters that is getting flashed. So in the oddball paradigm, so when he's focusing on that particular word, that will be evoking the P300 in his brain. So through the EEG acquisition. So when that is being used, we will be able, that will help in the speller. So patient will be thinking or he will be focusing, he'll be just looking. So for example, in a locked in syndrome, so wherein person will not be able to move, will not be able to communicate. So this is an assistive device where they will be able to spell. So that will have huge implication in ICU or persons with brainstem stroke or locked in syndromes for assistive technology, as an assistive technology.
Yeah. So my question was, I mean, I think you answered it, but my doubt was that P300 is more of the novel stimuli, right? The oddball or whatever you mentioned.
Yeah.
So what was the oddball thing out here? What was the novel thing? Because spell check, what you want to, you're looking for something. There's nothing novel out there, but I think there is a lot of letters out there, and
yeah, it will be the frequency. It will be the frequency of the rare and the common letters or the words that are being projected.
Okay.
So that is the target and the non-target words or the letters. So that is how. So when the person is focusing on that, then that will elicit the particular frequency in the brain.
Okay, great. Okay, another question, any work at NIMANS with this kind of setups?
So P300, not yet. So not yet started, but I'm really interested and I want to look into it. And there are research grade EEGs. So that is one thing. Then portable EEGs where we'll have to try out all these. And then most of these are available only in the English literature. We need something in the local Dravidian languages. So there are so many other things which needs modification. Culturally relevant modifications are required. And but the one which I was talking about, motor imagery BCI, that we have done work at NIMHANS in stroke subjects with IIT Palakkad collaboration, and the other one which I showed was at University of Houston. So those are some of the preliminary work which we have carried out and published, and I'm interested in taking that forward in neurological disorders.
Okay, great. Thank you.
Thank you.
Thank you very much. So how are you doing about time? We are quite good about time. Any more question? Well, we can still have one more question, and then if not, then we'll go to the next session.
So I have one question
please.
Professor Mitali. So basically working on Ayurveda and AI. It is a combination of Ayurveda and AI. So is there any method which is given in the Ayurveda for the treatment of cancer that we can apply, onto which we can apply AI and come out with some interesting things?
So Ayurveda, as you know, is very, very ancient, and they describe diseases on the basis of phenotypes, which is what we can connect with the modern perspective. So for example, if cell proliferation is a phenotype, Ayurveda describes that as a perturbation of the Vat Prakriti, okay, meaning Vat dosha. So now what we have done, there's a paper which has come in Cell Cycle recently. What we had done was, from the various extreme constitution types of Ayurveda, which are described as Vat, Pitta and Kapha, we made lymphoblastoid cell lines and tried to see whether they have a cellular phenotypes. So when we actually grew them into in the lymphoblastoid cell lines, we found that Vat, the cell lines which are derived from Vat have more higher cell proliferation rates compared to the Kapha. And this we had taken multiple biological replicates and technical replicates of each cell line. And what is very interesting is it is described in Ayurveda that processes like cell proliferation and all are expected to be higher in Vata.
Yeah.
Now, this was like a little contradictory, because these individuals who are of Vata phenotype, they have a very lean body frame, whereas a phenotype have a bigger body frame, and it might be anticipated that cell proliferation could have led to bigger body sizes. But here we see that cell proliferation were present in people who had phenotypes of a smaller structure. Then we tried to ask what will, if we give a DNA damaging agent to these types, would they respond similarly or to same extent? And what we found very strikingly is that if we give UV, the ones which proliferate faster actually has more apoptosis. But if we wait for some time, then they actually, because of their inherent cell proliferation rates, they become similar to the other one, where the killing is less, which tells us very important thing, that if we are doing precision medicine and if we are looking at the cell proliferation as an end phenotype and calibrating, let us say, dosage of UV, if there are inherent differences in such kind of processes which are also relevant in cancer, there might be differences in response to in recurrence or some such thing. So we also find inflammation processes to be differing between them, or hypoxia response differing between them. So the endophenotypes might be different amongst people, which might also lead to differences in outcome or pathways in cancer. So subphenotypic cancer might become very better if we actually conditionalize it by the Prakriti thing. So that is where we have got one paper where we have actually used AI, ML, and lasso to be able to see whether if we do an unsupervised, so we just take the questionnaire attributes of these constitution types and try to do unsupervised clustering. We try to see whether they actually have differences in their, they map to different clusters, and whether we can actually then start to use objectivity in the assessment. So that is what we had published long back in 2015. And that was the closest to AI ML I have done with my colleagues.
Yeah.
So this seems to be interesting. And only thing what is Ayurveda is very meaning subjective for us and not objectivized in the way we look at data. So if we want to do that, we have to connect it to a particular platform together. So molecular way is where we think we would integrate.
So I'm really great to see that the IITs is doing this kind of things. And one striking thing is that the precision medicine, which is the very hot topic in recent science, the Ayurveda knows this particular means to treat the humans since long with this precision medicine. It's quite interesting. Thank you. Thank you, Dr.
Thank you very much. Thank you very much for this live discussion. And now we move to our last session by Professor Stefani Alassoniere. Sorry if I pronounced wrongly, Professor. And she is from University of Paris, Peri-urban. And she is going to talk about 3 examples of decision support systems, outputs of my peri researches.
Thank you very much. So that's nice to be here, and I thank the organizers for inviting me to propose 3 examples of what I've been doing inside this Peri Institute. So Peri Institute has been created 2 years ago to gather together people from different background in artificial intelligence to support interdisciplinarity applications, and particularly I'm in charge of the health sector. So inside Peri, I'm also a researcher and a deputy director, but as a researcher, I will show you 3 different aspects of my researches. So the first one, can you see, oh, maybe it's not full screen. Can you see the screen now?
Yes.
Oh, great. All right, so the first one I want to mention is some research that we've been conducting together with the head of the maternity department in Necker Hospital. So this department is very famous for the analysis of fetuses that have abnormalities. And so half of the time is devoted to reinsurance. So basically, a mother are coming with a baby that has a small deviation with respect to some standard measurements. So for example, the baby has a too small femur, or it has an additional finger in the foot. And in most of the situation, this is completely safe for the baby. But sometimes this is linked to other phenotypes and correlate together. They are actually showing that the fetus have a rare and sometimes very severe disease. So half of the time of the practitioner in the maternity is devoted to reinsurance. Your baby is fine. Of course, this additional finger is not good looking. And we can think of surgery when the baby will grow and so on and so forth, but your baby is fine. So reinsurance. And the other half is trying to find what is the real pathology underlying the phenotypes that are showing up on this fetus. So we have developed like the ways of the echography, of the sonography. So basically you take the sonographic exam, you start from an isolated phenotype, which is different from the normality, and then ways, well, this is called sonio, will tell you what is the good way, what are the phenotypes to check to optimize your diagnosis. So basically, after having counted that there is an additional finger on the foot, then sonio will tell you, you would rather go and see what the heart is telling you, and especially if there is this phenotype or not. And this algorithm is always been updated real time all along the exam to produce a decision support system, which will either tell you, well, the baby is fine, there is an isolated anomaly which does not affect the health of the baby, or, well, there is an anomaly which is actually a syndrome that we have in our database covering 90% of the rare diseases on fetuses. And this is not still yet completely sure, but if you go and add, for example, a blood test or whatever tests, then we will be on 100% sure. And so you will have to go to your medical doctor and see what can be done for the baby. So this is based on reinforcement learning. I don't know how familiar people are with AI technologies in here, so I'm sorry if I'm too vague or too deep in the explanation, but this is based on reinforcement learning, where you have actually a random environment. So this is not like a chess game where if you move the piece, then the piece is moving. Here, if you go and move to see if there is an anomaly, then there may not be an anomaly, or even worse, you may not be able to see it because the baby is moving, because of the quality of the echogeneity of the mother, because of many other factors that does not help you get that information. So you are in a very high dimensional environment that is also random, full of randomness, and that you don't know much because there are actually very rare knowledge about the prevalence of each anomaly given the syndrome. So basically, sonio is really providing you the best path to go to do your medical examination, to get the reinsurance or the name of the syndrome as fast as possible. So of course, this avoids stress on the mother if there is nothing to say, or this enables to really make the mother go and see a specialist in that disease so that the baby can be taken care of. So this is really interesting because we were able to produce two software, which are Sonio screening and Sonio diagnosis. The screening is to help you doing the regular exam and being sure that in terms of quality control, you are of higher quality as possible. And the other one is Sonio diagnosis, where it really does either the diagnosis of everything is normal or the diagnosis of any of the 300 or 400 syndromes that we have in our database. So this was the first example, and this is based again on reinforcement learning and environment estimation. Oops, sorry. The other example is a close collaboration with Stanley Dorneman, who is a lead of an INRIA team in Institut du Cerveau, called Brain Institute in France, in Paris, in Pitié-Salpêtrière Hospital. So this is a joint work when we wanted to model the longitudinal data. So basically here, the data of one patient are given at several different times. For example, targeting neurodegenerative diseases, we have patients that have mild cognitive impairments that are coming to the neurologist, and they have several tests to perform. And so, for example, we have a person coming at 72 doing some tests, and then coming 1 year later because the neurologist thinks that there is nothing to do before, and then doing some similar test or some other tests, additional tests like an MRI, for example, and then that patient doesn't come anymore because everything is fine. Or we have patients that come at 60 and 60 and a half year old because the disease is progressing, and so on and so forth. So basically, we really model the evolution of the disease, trying to figure out if we can estimate from all those patients together that have different age, that have different start of the disease, although we don't really know when the disease is starting, and then different pace, of course, of evolution. All these people together are actually showing us the broad view of the evolution, the natural evolution of what people are suffering when the Alzheimer's disease evolves. So we have been doing this to really model the evolution of pathologies. We also did it for oncologists, trying to anticipate the time where the patients are actually escaping from a given treatment. A given treatment usually starts being inefficient and then starts being efficient at some point, and then so the tumor is decreasing, and then suddenly that you should think of changing the lines of treatment. And so we are trying to anticipate this before the tumor grows again, and sometimes the first part becomes completely erased by the new increase in terms of pathology. So here is an example of what we've been doing for the Alzheimer's disease. So the population came from an open access databases, which is called ADNI here, where we have many people that have been tested all along from 55 to 95-year-old. And so we were able, by conjugating all the information that they are giving us, to reproduce the evolution of the memory loss, concentration loss, language and practice loss all a long time for an average evolution of Alzheimer's disease. And I don't know how, if I have time, but I will skip this first and show you what it is used for. Imagine that we have these curves that I just shown you, and in a very quick example, I have a new patient coming into the neurologist room, and these are the score of that person. I personalize my model so that now I know approximately how these people will evolve along age, okay? So that person, for us, will have a diagnosis of Alzheimer's disease earlier than 80-year-old. Then the neurologist seems to think that this is not enough for him to say that this person is suffering from Alzheimer's disease. So basically, we try to wait and see what are the scores 6 months later for that person. That person comes back, and then again, our guess of diagnosis is earlier than 8-year-old. So still, the neurologist wants that person to come again another 6 months later and later and later. And finally, at 80, that person comes again into the neurologist room, and the neurologist does act that this person is suffering from Alzheimer's disease. So that person has been waiting 3 years before being diagnosed, although we were able to anticipate the diagnosis. So this tool is really used to anticipate diagnosis. Well, you will say that for Alzheimer's disease or any kind of neurodegenerative diseases, this is quite interesting, but there is no impact because there's no treatment. Although this is not completely true, because in July, Biogen proposed a new treatment for Alzheimer's disease, but maybe we have to think of something else. The treatment, all the 100 molecules that have been tested, at least in clinical trials from phase 1, 2, and 3, were not efficient. Maybe not because they were not efficient, but because of the therapeutic window was too late. And if we were able to anticipate, like for that person, 3 years earlier, the diagnosis, then we would propose that person to take the drug and see if that person reacts well and slows down the effect of the disease. So basically anticipating, even for neurodegenerative diseases, anticipating diagnosis is really important. So going back to the third example that I want to show you, I think I'm quite on time. Let's see, yes. So to show you the last example is concerning data augmentation. In all medical application, we usually face the small sample size of our data. We have to deal with, let's say, 50 data that come from a unique hospital, and we cannot make them go out of the hospital and share with other hospitals that would have kind of the same data. So basically, what we were recommending is to augment the size of the data artificially, trying to design relevant and completely synthetic patients that will share the characteristic of the initial population, but that will not be related to any other patient. Basically, that will be anonymized because they are pure synthetic and will be not able to go back to the patients with the synthetic data, so that these data will first be of a tremendous amount. So for example, here we have used 10,000 synthetic images that we have completely produced artificially to train a classifier, but also these 10,000 images can go out from the hospital, be joined to another 10,000 that are coming from another hospital, and construct, let's say, more representative database of what happens in the area of interest of this disease. And so we've been creating algorithms to produce images that are really relevant. So the top line are patients that are considering as normal, although here there is a patient that is quite old with big ventricles. And the second lines are persons suffering from Alzheimer's disease that are relevant with respect to a radiologist and a neuroradiologist eye. And we've been testing the relevance of this augmented dataset with respect to the classification on the ADNI database, same, and the other database, which is also open access, which is called ABLE. It's the same protocol as ADNI, but ADNI is North American, and I think I would even say just US database, and the ABLE one is for Australian patients. So it's the exact same protocol, so we barely have the kind of same images, but basically we're training our algorithm on the ADNI database, trying to say if we increase the classification power on the Australian population as well, knowing that the Australian are not too far from American, that's for sure. But what we can see is that for all sensitivity, specificity, and balance accuracy, we were able with augmentation to outperform the classification score even on some population that we've never seen in our population, initial population, so our training set. So basically, what does it mean? It means that we are actually capable of capturing first the variability in the real image that really encodes for that classification. And we are also to generalize well, because we are able with those synthetic images to outperform classification on images that are of the same kind, but on another population. So these are the three subjects that I am still conducting in the Prairie Institute. And so one is now startup, Sonio, and the two other are ongoing. Thank you. I hope I didn't take too much time.
Thank you very much, Professor Stefani. You did a fantastic job. 3 different problems, all challenging, and you provided very interesting solutions to that also. So now I will open the floor for question. Actually, we'll have 2 parts since all talks are over. First, we'll take questions specific to your presentation. And after that, of course, we'll have a general discussion. So any question from the participants, speakers?
That was a very nice talk. And as a cognitive neurologist myself, I'm also interested in the work that you had shown. So in the prediction model to Alzheimer's, so was it only the clinical parameters, the language, attention scores, or was it also combined with the imaging parameters?
So the plots I showed you were only estimated using the scores, the neurodegenerative scores. However, we did the same kind of analysis on images, which are MRI,
okay,
which are PET, and we had also segmented the hippocampus on the shape of the hippocampus. So we were able also to draw the evolution of each. I can probably share my screen as well and to show you what are the results for that. Give me 1 second. And I will just show you exactly what we finally end up with. Share screen. Where is share screen? Here. Share screen. All right. So basically, here is it. So basically we had everything. Oops. We had the cortical thickness, the glucose consumption, and the atrophy of the hippocampus with respect to the cognitive scores.
Okay.
We did it on the, let's say, 4th modality.
Okay, thank you.
You're welcome.
Any other questions?
And do you think this can be extrapolated to the other dementia disorders? So we get to see a lot of young patients with dementia, say, for example, the frontotemporal dementia. And then in India, we get to see a lot of vascular dementia. We see different pathologies and MRI showing significant white matter disease, and sometimes Alzheimer's combined with vascular dementia. So we get a range of such disorders. Do you think AI and application of the same model to these disorders also can help in predicting the outcomes?
Yeah, so this has been tested on Alzheimer's, of course. We have been testing it on Parkinson. We have tested it on Huntington disease as well. We were not able to have young people with other diseases because we are, well, we highly depend on the databases that are shared with us. And so in the Brain Institute in Paris, we had a chance to have experts for Parkinson's and Huntington in addition to Alzheimer's, but we were lacking other expertise.
Okay. Right. That will be very interesting to explore as well.
Sure.
Thank you very much. Any more questions? If not, then I will ask some questions. The first one is about your Sonio diagnosis. it seems it's already being used, right?
Yes, it
So,
is.
and it's not only, I mean, it not only detects the abnormalities, but also it classifies, right? Since you say diagnosis, I'm curious because when it comes to disease diagnosis, to me, it looks like an open world problem, which means that we may train our system with 10 things, but there may be an 11th thing waiting for us, which we have not seen, or it's extremely rare. So that's in terms of AI language, we call it really open world, and most of the time we design system as if it's a closed world. So, and it's very important for medical diagnosis. So in your system, I do not know because I'm not an expert in the field, but suppose it is ready for 100 types of abnormalities. And if 101 type abnormality comes, what will it do? Will it assign to one of them, or it will say something like, I am not in a position to make a conclusion?
So we have exactly what you say. For example, if you consider the rare diseases,
right,
you have 7,000 of them.
Right.
However, the amount that you can diagnose use in the fetal part of life,
right,
with ultrasound reduces to about 500.
Okay.
Okay. We have covered, I think, 350 of them that are the most, the less rare diseases. We are only dealing with rare diseases, so basically less rare. So it covers 90 to 95% of the possible diseases. Of course, at some point, if the baby has something that is not in our database, with specific phenotypes combined together that are not something that we have trained our algorithm to recognize, it will just say that it's something out of our database. But then the medical doctor just have to open a book and see in the possible diseases that remains, about 100, what is the one that could happen to be. The interesting thing is that if that happens, we have a loop back to
Correct.
our system, which says, oh, by the way, you forgot one thing. And so we will retrain our algorithm with a new environment, including that. At the end of the journey, we would like to cover 100% of the disease, which will not be possible because we never know 100% of the rare disease. New diseases are just showing up anytime. So basically, we will cover, let's say, 98%. We hope in a near future to cover that amount of diseases.
Fantastic. This is really nicely done, which most of the AI system often don't do. I have another simple question. In your longitudinal data analysis, it seems you have taken care of several diseases, and I understand it's pretty domain specific. So is it a framework which can be adapted for any disease? Like I have degenerated discs. So can this be, if you have data, it's a framework which can be adapted to it?
Yeah, this is completely blind to the disease. Completely blind to the disease.
Excellent.
And if I can show you just one slide to show you how this works, it works in terms of math only. So we are considering that the data are living on a Riemannian manifold, telling that each data is just some equation related to the natural history of the disease that we are estimated with some variability. And I don't want to go further in terms of math, but basically think of any kind of disease you want to draw the evolution of. And you think that patients are deviation in space, meaning in anatomy, and in time, meaning of pace of this trajectory. So that the average population, a random characterizer of a population would walk on that trajectory, basically something that an atrophy of the hippocampus. So you will have here the orange curve will be the atrophy of the hippocampus along Alzheimer's disease. And then each person has its personal shape of hippocampus and a personalized pace of atrophy, which is considered. But you can think of oncology, evolution of a tumor. You can think of any kind of evolving pathology that you have data and you can model them with respect to that mathematical background, then everything comes into the same model.
Correct. This idea of manifold for this disease samples lying on a manifold suffering from a particular disease is fantastic, and it makes perfect sense. I'm sorry, I have the last question. This is about the synthetic images. So you generated really very interesting images, but do you take into account the biology, physics, stuff like that into it, or just the everybody does this image, what they do in image processing?
No, we don't put biology constraint. That's correct. We only take the images, put them in an variational autoencoder that has its latent space that is modeled again as a Riemannian manifold, where we estimate also the Riemannian metric. So basically the data are informing the encoder, decoder, and the Riemannian metric in the latent space, but everything is driven by the data, except the shape of the metric, which we reduce to something that is parametric so that we can optimize with respect to the parameter of the metric. Otherwise, we won't be able to do some optimization. So the metric has some simple shape that we are dealing with to estimate like a landscape of mountains and valleys in the latent space, but otherwise everything is driven by the images themselves.
Fantastic. I have no more questions. If anyone, I'm sorry that I asked too many questions.
No problem. Thank you.
Anyone else? If not, then we'll move to the second part, which is very important. Jan, I believe you are also there, and I would invite you to comment on this also.
Yes, so, I'm here. I'm very sorry because I have some trouble with internet, so that's why I'm connected in 2 places. So I'm sorry, but I won't be able to interact easily.
No problem, no problem. I gave you enough trouble this morning. Don't worry. So basically, what is the objective of this thing, this part? First of all, I would like to thank all of you for these excellent presentations, and my expectation or hope is that some of you have already found out, like your partner from the other country, to discuss for future potential collaboration. If that has already happened, it can happen in near future also. Then one part of this 2-day workshop is certainly successful. Secondly, for this discussion session, we don't have a predefined agenda. Whole idea is to come up with some suggestion, some recommendation, maybe a list of actionable items, which we can submit to, I do not know, we'll submit to the French consulate probably, but this will finally may go to appropriate people of both countries. So with this objective in mind, may I invite the panelists are all the speakers, even from the audience also, if they can come up with some suggestion, this is the area we should focus on, or this is the problem we may like to have a workshop next year, something like that. So any volunteer to begin the show? Dr. Rahul, would you like to?
Yeah, so my focus is to have hands-on training, I mean, on physical mode. If you can look the things, and it will be better, I guess. So we can think about that as well.
For the next edition of this.
Year, just like a workshop, hands-on training kind of things, so that we can imagine the tool, we can see the tools, we can operate the tools at least to some extent, so that we have a more precise and meaningful idea, it can be converted into something very, which is very nice.
I fully understand that hands-on training is much more effective to get fruits out of any workshop. That's true. I have noted it down, and this would be a point which we'll try to mention.
And importantly, here in India, we have so many centers for artificial intelligence and data science, as Professor Mitali said, right? One of the center is in IIT Jodhpur, so we can collaborate with them and we can take it to the next level via physical mode.
No, here what I am referring to is this is under the Indo-French framework. So we can certainly, like in ISI, we have a center, we have organized a couple of programs, we had hands-on training also. But here our objective is to develop something jointly where both India and France get benefit out of it.
Yeah.
But that's a good suggestion. Even under this Indo-French framework, we can extend it to including hands-on training also.
Yeah.
Very good. Thank you very much.
So I have a question, Dr. Pal.
Yeah.
So yes, some of us may, for example, want to go back and look at all the talks and figure out the synergies. So will it be possible to share some of the recordings or the recordings that have been made for the two-day program?
I cannot comment on this, but I don't find any reason why this cannot be shared, because some of the speakers have already said that they can share their slides also, and I think it's a good idea. What you said, I will take a note of this. Sharing of the videos. Yes, I mean these are all scientific discussions, so. Personally, I don't see any issue, but however, there may be issues that I cannot see, like copyright or something. We may have to get permission from every speaker, but I'll leave it to the main organizer, which are much higher level than me.
Thank you.
Thank you very much. This is a fantastic idea, and this can really lead to successful collaboration between the two countries. Yes.
Yes. I would also second what Tapritesh said just now. That's because I would have lots of other colleagues who might be even more keen to have some synergies, which we can take it up. For example, in the area of cancer, we are trying to develop a program on radiogenomics because we have strength of computer vision and linkages to cancer hospitals and even the genomics. But then this could be actually the methods and the data can be compared jointly between the centers and things like that. So I would really like to second what Taf said, that if we actually get to circulate the link within our colleagues in the aid school, let us say, then that might actually lead to much better synthesis of what are doable, right? So from our institute, I can say, if we are talking about this, there's a lot of expertise being developed on phenomics, meaning in the sensor development, and also trying to integrate multiple levels of data for precision intervention, identifying predictive markers, and the desert medicine, that how you look at signatures of selection in extreme environment and integrate it to medicine. So, so anybody who would in India or in France who might be wanting to get to that, we might actually posit this as problems from IITJ. We would be keen. So that is something which I could mention from our side.
Thank you very much. Any other suggestion? Like something which we can propose to the Indo-French organizer, the CEFITRA. Let me put some random thoughts. This may be totally useless, but just to carry forward the discussion, because both yesterday and today in several of the talks, those who deal with real healthcare data, they have emphasized directly or indirectly that there are many issues with real healthcare data. Even in hospital setup like in India, I know that if a patient gets admitted, there may be some records available. But if it's an outpatient, that means the patient comes and gets examined by a doctor, and doctor writes a prescription, and often there is no record available. And also there are issues of missing data, etc. So health data science, I think this is the term used by Professor Sethi. I like that very much. So do you think that this is a good idea to develop some guidelines, tools, and techniques for real-time capture, creation, management, annotation, privacy preservation, and exploitation of health records? What I'm trying to refer to is some kind of standardization in the context of collection, maintenance, and use of healthcare data. Because in the era of AI, as you know, that standardization has become a big issue. And I can produce a car with an autonomous driver, autonomous car, driverless car, and the norms that I have followed may not be acceptable to some other country where I try to send the car. So some kind of standardization in terms of some kind of guidelines to enforce the standardization or develop the standardization. And as well as you need some tools and techniques also for really creation of the healthcare data, because these can help a lot, whether you try to develop infrastructure for the country and whether you try to kind of mine the data to come up with something novel. Does it make any sense, or it's totally useless?
I completely agree with you, Professor Pal, and I think both for the India and French side, I think as we realize more and more that data is key for training good models. And as you said, electronic health records are just taking form in India. The recently announced National Digital Health Mission, which is going to go hopefully go big scale in the coming years, has given, I think, some frameworks. And we have used some of these frameworks in our Delhi cluster project. Dr. Mitali Mukherjee has been a part of that, where using the standardization frameworks was a key aspect to help data exchange happen for COVID scenarios. Now going forward, we believe that COVID was just a testing ground. But if we could, for example, come together with French colleagues and figure out what are the best practices where data, even if not data exchange, I know that health data are extremely sensitive, but there's a lot of approaches available. For example, synthetic data generation from available data sets, which can then be shared, federated learning, federated artificial intelligence. So all those, as I said yesterday, I think certainly I would be very much keen to discuss with French colleagues on demonstrating some use cases or studies that can enable these. Would be happy to discuss.
Thank you very much for this useful comment. And I did not know that there's some initiative, at least keeping in view the COVID-19 data. Thanks for this very useful comments and some idea to move way forward. Any other suggestion, comments?
If I may, would it make sense to organize some kind of a summer school for the PhD students or the graduate students we have in our different labs, and put everyone together, in broader scale than just some representative of the labs right now? That would be a way to have a kind of a hands-on approach, as it was suggested before, and to go more in depth into what we are all doing.
Fantastic. Summer school or workshop involving. So this will involve the research students also, master students, graduate students. Yeah, very good idea. Very good idea. And I must agree with you, like every talk had a lot of content, but I really felt ashamed that we gave only 15 minutes time because each of the topic that you all have talked about probably can take more than 15 minutes. So this is a good idea. So which can include also hands-on. Thank you, Professor Lekunov. Thank you very much. So, this is also a concrete idea that comes up. Any other suggestion?
Sir, we can have some kind of collaborative projects between universities, or we can say between continents, so that again it can be a collaborative learning among the universities and the faculty members, or some kind of faculty exchange programs can be organized. So
okay, let me just add here a little bit, because one of the objectives of this workshop is that after we hear each other, we'd be able to find my closest partner in the other country, and then we may think of writing collaborative projects. Now, CEFIFRA, the Indo-French Institute in India, they provide some fund, and they every year, I don't know how many times they float the call for proposals. In fact, one of the participants had already a project under that scheme. So this is a good idea, and that in any case will be one of the byproducts, so that collaboration between scientists from the two countries can be initiated. But you have added something more, that collaboration between universities. So do you have anything beyond this in mind?
Yes, sir. Like in NIMS, it is a DIMDB university in India. We have one kind of like a scheme, like in which we can go for collaborative research projects. And but we need to check it, like other universities are open to do this or not, so that if it is like available, then we can sign an MOU between the universities, so that we can say the fraternity of research and innovation can be added together. I would like to add one more thing, like we can initiate some kind of, or we can promote some kind of startup research grants, so that it will help the early age entrepreneurs to the field of AI with the, we can say, this healthcare industries. So
Okay.
like so this thing can be like we can say taken place in the next level of handling healthcare related services and support across the countries.
Okay, good idea. So I am taking note of these things, so we'll include that part of our recommendations and suggestions from this from this 2 days deliberation. Thank you very much. We still have more time. Any more suggestion, question, issues to discuss?
Just a question, maybe, because I completely agree with the idea of sharing the talks, so that we have time to look at them again more, slower, let's say, and get all the information. But then what would be the follow-up from this? So let's say we look again the talk, and then we realize something could be done with, jointly with someone who presented. So maybe, okay, so I guess it's a question and a suggestion as well. Should we organize some sort of follow-up, or do we leave it to the people to contact themselves how they wish?
The impression I got is this, that once we know each other, we find our closest researcher who may be interested, and then they directly write to each other, and then work together to develop proposal. And when this Indo-French call for proposal comes, it is submitted under that. But I also, as I said, that I also agree with you that if I could get the videos, first of all, I have missed several talks today because of power cut. So and otherwise also, if I get the videos, I myself, irrespective of whether I go for project or not, I like to listen to them slowly and take pointers and go further on that, because the talks were very interesting. Thank you very much. This is a very useful idea, and it emphasizes this fact that we should share the talks.
Yeah, I have a suggestion, like,
Please. We cannot hear you.
Yeah, I cannot start the video. I mean, it feels like the video button is not working. Okay,
This-
whatever, that doesn't matter. So I mean, although I was not a speaker, but it feels like, I mean, if we can identify some sort of key areas, like key challenging areas of AI and healthcare, from the discussions that we have in the last 2 days, that might be a great thing to point out. I mean, what could be the key focus areas of research as this collaboration between India and France? So are we able to figure out some 2 to 3 key research areas that might be more challenging and more people are really interested into that?
Well, that was one of the objectives.
I mean,
also
like a thematic issue, what I mean.
Right. So it's kind of recommendation that this is an area where we should focus. We should emphasize more on this, something like that.
Just got the idea, like, I mean, if a project proposal can be announced that, okay, this is the thematic area, and if people are interested, they can apply collaborative projects.
No, usually
so, I don't know.
if we recommend some area, then there is a chance that the higher level body will take into account that when they put the call for proposal. But we have to suggest something like that.
Exactly, that was my point. If it can come out from the discussion, from the speakers, that will be great.
For example, what came to my mind while going through this discussion is lightweight, less data hungry AI ML algorithms for healthcare. Of course, if it is lightweight, the chances of making it maybe explainable is high. Now, this is an area I thought could be a good area to focus on, or even initiatives for affordable rural healthcare. Now, this is of course a big project, probably it's the local government should take care, but lightweight, less data hungry AI ML algorithms. Personally, I was thinking that this is something very important for many applications. We may not have good amount of data available in a form that can be used. And of course, we get sometimes once in a while crisis like COVID. So if anyone has any suggestion on this, some areas where we should focus, of course, it should have some challenges, then we can include that as a part of our recommendation. Many of you are working directly with healthcare data, so I'm sure you have faced a lot of problems while coming up to these nice results. So if you can. So it seems that we may not get, well, we still have few minutes time, so we can still wait. And if you otherwise, we may.
I just have a small comment to make that it's a wonderful workshop, and as a healthcare provider directly dealing with patients, especially at NIMHANS, which is an institute of national importance, where we have all the resources and all manpower, including people from neuropathology, neuroradiology, neuropsychiatry. I'm open to application of AI and technology in patients. So if anybody wishes to have some collaborative work where patients will be benefited, and the technique or a product can be applied, a simple portable one, less costlier one, which can be applied on patients, then I'm more than happy to be part of it. And I will be able to help and improvise on the data or the patient recruitment and the project and other things. I'm happy to help in that way. I just wanted to add on.
Thank you very much. This is really a very useful open invitation, because I know the place you work on, how important it is and how much access to real life data that you have, and how complex and difficult patients that you need to handle there, and their volume also. So that's a thank you for this invitation, and I am sure that you will hear from some of us definitely. Who knows, I may write you soon. So
Thank you.
it's
Thank you.
most likely some of us will.
Ma'am, can you please share your email ID in the chat box so that
Yeah.
we can reach you?
Because in India, it's very difficult to get doctors who are willing to spend time on this kind of research. So it's, thank you very much, ma'am. Thank you very much. Any other comments, suggestions? So friends, it seems there is no more question, comments or suggestions. We are supposed to close at 5, but we can close at 4:57. Nobody is going to say anything for that. So before I conclude, I would like to thank all of you for your presentation, those who have presented an active participation. All the talks that I could listen to, I have learned a lot. Maybe I couldn't follow everything because there are a lot of materials, and we forced you to squeeze in 15 minutes time, 13 minutes time, and so it was difficult. But still, I must say that I got a lot of things out of this workshop, and I hope that others also got benefited out of it. Thank you very, very much. Thank you very much.
Thank you. Thank you so
with
much.
this note, we'll close the session for today. And everyone have a
Thank you,
great
sir.
day.
Thank you, everyone.
Thank you.
Thank you.
Thank you very much.
Thank you.
Thank you. Bye-bye.
Everyone, thank you.
Bye-bye.